Good morning, students. Today we are continuing with this chapter, Sexual Reproduction in Flowering Plants. In that chapter, today we are discussing post-fertilization, in that structure and events. So what are the structures involved in post-fertilization and what are the events occurs in that we will discuss here. The post-fertilization events includes two steps, that is endosperm development and embryo development. Now, the maturation of ovule into seed and ovary into fruit means the ovule mature into seed and the ovary is mature into fruit. The primary endosperm cells, yesterday we discussed that one. The primary endosperm cells divide repeatedly. The primary endosperm cells divide repeatedly to form triploid endosperm tissues. They form the triploid endosperm tissues. Endosperm cells containing a reserved food material, large amount of reserved food material which is used as a nutrition to the developing embryo. This is the reserved food material is used as the nutrition to the developing embryo. The common endosperm development includes the PM, that is primary endosperm nucleus, undergo successive nuclear division to form the free nucleus, that is free nucleus are formed. This stage is called as the free nuclear endosperm. The primary endosperm nucleus undergo successive nuclear division. This will form gives rise to the free nucleus. This stage is called free nuclear endosperm. Remember it. Then the number of free nuclei varies greatly, means different in embryo will have the different number of free nuclei. Then the endosperm becomes cellular. The endosperm becomes cellular due to the cell wall formation in the endosperm becomes cellular because due to there is a cell wall formation around them. Then one of the example about that is the tender coconut water. The tender coconut water is the free nuclear endosperm. It is a free nuclear endosperm. And the white kernel is the cellular endosperm. It is a cellular endosperm. Here the free nuclear endosperm includes, they are made up of thousands of free nuclei which are moving inside the tender coconut water. So this is the one example about that. Next we discuss the second step. That is, first step is the endosperm development. How it is developed, we will discuss it now. That is, the primary endosperm nucleus undergo successive nuclear division to form the free nuclear endosperm. This free nuclear endosperm around that there is a cell formation is called cellular endosperm. Example, you can take the white kernel is the cellular endosperm, and inside that there is a water is there, that is the tender coconut water is there, that water is the free nuclear endosperm. So this is about the endosperm development. Then second step is the embryo development. So embryo develops at the micropolar end of the embryo sac, at the micropolar end of the embryo sac, where the zygote is situated. Where the zygote is situated, at that place there is a embryo development will be start. Now most zygote divide only after the endosperm, only after the formation of certain amount of endosperm in them. This is the adaptation. This is the adaptation to provide nutrition to the developing embryo. So the most zygote divide only after the formation of certain amount of, only after the formation of certain amount of endosperm. That is the adaptation to provide nutrition to the developing embryo. Then, though the seeds differ greatly, that is monocot and the dicot seeds, the most of the seeds are different. The seeds differ greatly, but the embryogeny, that is the early development of embryo, the early embryonic development or early development of embryo is similar in both monocot as well as in dicots. So though the seeds are differ greatly, the monocot and dicot, but the embryogeny, that is the early embryonic development, is similar both in monocot as well as in dicots. Then the zygote gives rise to, so here the zygote is formed at the micropolar end of the embryo sac, where undergo the embryonic development. First they divide into two cells, that is called as the proembryo. So the zygote gives rise to proembryo. They undergo continuous successive division. They form the first, subsequently they are undergo divisions to form globular embryo, then heart-shaped embryo, then mature embryo. The mature embryo, what it includes, they are including the radicle, the plumule, the cotyledons. What are the radicals? What are the plumule? What are the cotyledons? We will discuss in the first year only. So what is radical means? This is the part from which the root is originated. Plumule, this is the part from which the stem is originated. Cotyledons, these are the first two leaves of the developing embryo. So this is about the how the endosperm development takes place and how the embryo development takes place in the as in monocot as well as in the dicot seeds or the dicot embryo. So here the endosperm development, we will discuss the primary endosperm cells. divides repeatedly to form the triploid endosperm tissues, and the endosperm cells containing the reserve food material, large amount of reserve food material which give the nutrition to the, or which is utilized to the nutrition to the developing embryo. Means endosperm is important for the embryonic development. So here the after the formation of endosperm. So this endosperm includes the common endosperm development includes the primary endosperm nucleus. Earlier we studied the primary endosperm nucleus undergoes successive nuclear division to form the free nuclear endosperm. That is, they form the free nucleus. This free nuclear stage is called as the free nuclear endosperm. Then the endosperm cells are formed. They are formed cellular level. Why? Because due to the formation of cell wall around them. There is a best example is the tender coconut water is the free nuclear endosperm, and the white kernel around them, that is the cellular endosperm. So now the embryo development includes the zygote undergo development at the micropellar end. So zygote undergo development which is situated at the micropellar end, and that zygote is formed. Development takes place only after the certain amount of formation of the endosperm, because this is the adaptation for the giving nutrition or providing nutrition to the developing embryo. Then though the seeds are differ in structure, but they undergo similar embryonic development, that is embryogeny, that is early embryonic development is similar both in dicot as well as in monocots. So this is about the stages occurs in the embryo development, where zygote undergo division to form the pro-embryo, that is two-celled stage. That pro-embryo undergoes successive divisions to form globular embryo, then heart-shaped embryo, then mature embryo. In mature embryo, there is a formation of radical, plumule and cotyledons. So plumule, this is the two cotyledons, means this is the stage in embryonic development of dicots. Okay. So radical is the, radical is the part from which the root is developed. Plumule is the part from which the stem is developed. Cotyledons, these are the first two leaves in dicots embryo. So this is about the steps involved in the post-fertilization events, that is endosperm development and embryo development. Now we discuss the structure of dicotyledonous embryo. We can draw the diagram of a dicot embryo, a typical dicot embryo diagram. Then we discuss the structure, what are the parts involved in their structure, that we will discuss here. Before that, you should remember that the embryo development, the stages in the embryo development of dicot, the diagram as well as the explanation carries 5 marks, and it is very important for you students. Then we discuss the structure of dicotyledonous embryo. That is development, this is structure. So it is also very important for 5 marks. So structure, we have to draw the structure of dicot embryo. Now we label it. This is the plumule. This is 2 cotyledons. This is DICOT. This is hypocotyle. This is root cap. Root tip. It is commonly called as radical. It is commonly called as is commonly called as the shoot tip or stem tip. Okay, so this is about the structure of the dicotyledonous embryo. In the structure of dicotyledonous embryo, the dicotyledonous embryo has an embryonal axis. It contains the embryonic axis. This is the embryonal axis. In that embryonal axis, there is a 2 cotyledons are present. This is the embryonal axis where the 2 cotyledons are present. These 2 cotyledons, in that above the cotyledons there is the portion of above the cotyledons in embryonal axis is the epiphytile. That is from the in the above cotyledons on embryonal axis there is a part is present that is called as the epipodyle, and it terminates, this epipodyle is terminates with plumule or stem tip. So in the dicotyledonous embryo it has the embryonal axis. These embryonal axis, over the embryonal axis they have two cotyledons. The two cotyledons, above the two cotyledons on embryonal axis there is a part is present that is called the epipodyle, and that terminates with the plumule or the stem tip. Then the below the cotyledons is swelled the cylindrical part. This is the cylindrical part. So the below the cotyledons a cylindrical part is present that is called as the hypopodyle. So in the embryonal axis, on the embryonal axis below the cotyledons a cylindrical part is present that is called as the hypopodyle, and this hypopodyle is also terminates with radical, or it is also called as root tip. The root tip is covered by root cap. Remember the structure of dicotyledonous embryo. You have to explain the structure of dicotyledonous embryo with neat diagram for 5 marks. So here this structure of dicotyledonous embryo includes the embryonal axis. On the embryonal axis they have the two cotyledons. On the embryonal axis above the cotyledons there is a part is present as epicotyle, which terminates with stem tip or plumule. Then the on embryonal axis below the cotyledons a cylindrical part is present called hypopodyle that terminates with radical, which is also called as root tip. The root tip is covered by, it is protected or covered by root cap. So this is about the structure of dicotyledonous embryo. Tomorrow we discuss the structure of monocotyledonous embryo and the seed formation and fruit formation. Thank you.